Okay, class. Good day, and thank you for attending our laboratory today. So, for our meeting, I'm going to explain to you Activity 7, which tackles about the gram-positive biochemical tests. So, this test class are used to identify specific gram-positive cocci and your bacilli. So, let's discuss them one by one. Now, let's start with the first and the most common test for the group of gram-positive cocci. We have the catalyst test. So the catalyst test is a routine test done in almost all laboratories. Now the principle here is that during aerobic metabolism, some bacteria reduce oxygen and produce toxic byproducts such as your hydrogen peroxide and superoxide. Now these two, itong H2O2 and superoxide can damage cellular components of your bacteria if not removed. To protect themselves, many aerobic and facultative anaerobic produce the enzyme superoxide dismutase and catalase. So when certain bacteria class are exposed to hydrogen peroxide, superoxide, they produce these two enzymes to protect themselves. Now, when these two enzymes are produced, superoxide dismutase would convert superoxide into hydrogen peroxide. Then catalase would break down hydrogen peroxide into water and oxygen. And remember, when you mix water and oxygen together, you tend to produce bubbles. Specifically, when oxygen is released, visible bubbles is formed when hydrogen peroxide is added to a catalase producing bacterium. Thus, bubble formation indicates a positive catalase reaction, while no bubbles indicates a negative reaction. To summarize, class, the reagent that you're going to use here is your hydrogen peroxide, and the positive result would be the presence of bubbles. So to explain further, here's the procedure. So for the slide test, place a clean glass slide on the work surface. Then using a sterile wooden applicator stick or a nichrome wire loop, transfer a small amount of bacterial growth. So if this is your slide, you add here a colony, you mix it. Then once done, you add 1 drop of hydrogen peroxide. Observe immediately for the release of bubbles. If may bubbling, positive. If no bubbles, negative. So this is the image. So you get a small piece of the colony, you add it on the slide, then you add your 3% hydrogen peroxide. Again, if positive, bubbles would be present. If negative, no bubbles. Important. So this is very important, class, in identifying specific bacteria, mostly gram-positive cocci. So it is used to differentiate Staphylococcus, which is your catalase-positive gram-positive cocci, from the Streptococcus species and Enterococcus species, catalase negative. It can also help in separating gram-positive bacillus from other bacilli, katulad ni Bacillus species class, which is catalase positive, while another group of gram-positive bacilli, Clostridium, is considered catalase negative. So I want you to remember, class, to differentiate staph species from Streptococcus, it's always catalase. So let's continue. Let's go to the second common test for gram-positive cocci, the coagulase test. So this is coagulase is also an enzyme class produced by the Staphylococcus species. It converts fibrinogen in plasma to fibrin, resulting in clot formation. This clot protects bacteria from host defenses by surrounding them with a fibrin layer. So certain bacteria class, specifically Staphylococcus, would produce coagulase. It converts fibrinogen in plasma to fibrin, protecting them. Now, the presence of coagulase is strongly associated with a pathogenic species, specifically Staphylococcus aureus. A positive test is indicated by clot or clumping in plasma, while a negative test shows no clot formation. So, ang reagent nyo dito, class, is plasma. A positive result is clumping or clot formation. So to further explain, let's go to the procedure. So there are two methods here, the slide method and the tube method. Now take note, class, The slide method is known to detect bound coagulase, also known as the clumping factor. So here's what you're going to do. Place 1 drop of sterile saline or plasma on a clean glass slide. Emulsify a colony of the test organism into the drop. Then observe for clumping within 10 seconds. So here's your slide. You add here first your plasma. Then you add your colony. Emulsify it, mix it with the plasma. Then observe for clumping within 10 to 15 seconds. It's positive if there would be visible clumping, meaning bound coagulase is present. Negative if there's a smooth suspension without clumping. Now here's a note, class. In instances or wherein there are cases that the coagulase slide test is negative, you need to do a confirmatory test. So let's say you had a result that were in the negative, wherein your slide method is negative. You need to do a confirmatory tube method since some strains may not produce bound coagulase. So let's go to the method, yung tube method. So here's the appearance, class, of your coagulase positive. So if you take a look, may mga clumping, visible clumping is present on the coagulase positive, while no clumping is in the negative. The white here, yung nakikita niyo, those are the colonies. So there's no clumping. Basically, emulsified pa din siya. Now let's go to the confirmatory, the tube method. So this detects free coagulase. Adds 0.5 ml of rabbit plasma into a small sterile tube. Inoculate with several colonies of the test organism. Incubate at 35 to 37 degrees centigrade. Now you're going to observe this class in a pattern. Observe them at 1 hour. So at 1 hour after performing the test, check if there's already clumping. If wala pa, go for the 2nd hour. If wala pa din, go for the 4th hour. If negative pa din on the 4th hour, continue up to 24 hours. Any degree of clotting from a loose clot to a solid clot is considered positive, while a negative, no clot formation after 24 hours. So the tube method, class, detects free coagulase. Si slide method yung is yung clumping factor or the bound coagulase. So do remember that. So here's the coagulase test. So if you take a look, class, there's clumping na siya. There's a clot formation on the letter B slide. This is considered positive. Now the importance is that used to differentiate pathogenic from non-pathogenic staphylococci. So kung ang catalase nyo, if your catalase is used to differentiate staph from strep, si coagulase nyo naman is used to differentiate S. aureus from other staph species. Specifically, staph aureus from other staph species, yung mga coagulase negative. So this has been discussed in your lecture class. The most common is yung Staphylococcus saprophyticus, something like that. So differentiates coagulase positive Staph aureus intermedius lugdunensis from coagulase negative Staphylococcus epidermidis, Staphylococcus saprophyticus, and most other staphylococcal species. Now let's go to the third test, yung DNase test nyo. So the DNase test detects the enzyme deoxyribonuclease. Now si deoxyribonuclease nyo, or the DNase enzyme, hydrolyzes DNA into smaller fragments. Many pathogenic bacteria, especially staph species, produce DNase to degrade host DNA, increasing their virulence. So when bacteria producing DNase grows on a medium containing DNA, the DNA around colonies, class, is broken down. So you would be using here your DNase agar, which contains DNA. Now if you inoculate a bacteria that produces DNase, class, the DNA in that agar is broken down. So after flooding the plate with 1 normal hydrochloric acid, the degraded DNA dissolves, creating a clear zone around the DNase positive colony. Colonies without DNase remain surrounded by opaque medium since intact DNA precipitates in the acid. So ito yung procedure niya. So you're going to divide the DNase agar into sections using a pencil, label with your name and test organism. Heavily inoculate the surface of the agar with the test organism. Specifically, examples are Staph aureus, Staph epidermidis as control. Incubate the plate at 35 to 37 degrees for 18 to 24 hours. After incubation, flood the agar surface with 1 normal hydrochloric acid. Positive if there is a clear zone around the colonies indicating DNA hydrolysis. Negative, no clearing, medium remains opaque around colonies because again class, the DNA precipitated because of the acid. So pag nilagay yung, if one normal hydrochloric acid is added and bacteria does not produce DNA would precipitate. But if it produces DNase class, the DNA is destroyed and it becomes a clear zone. So ito yung itsura niya. So if you take a look at this first one, ito yung colony. So after growing it for 24 hours, you add the hydrochloric acid. If there's no clearing around the edges, negative siya. But if you take a look at this, if you take a look at the surrounding of the DNA, ito yung edge niya, di ba? The edge of the colony. If you take a look at the edge of the colony, you would see a clear zone surrounding it. So this is the clear zone. This signifies that this is the colony here is DNase producing. So ito yung clear zone niya. This is the clear zone. Now, importance is used for identifying pathogenic staph, specifically Staph aureus. Used to differentiate when coagulase results are uncertain. Useful in clinical microbiology to confirm virulence-associated traits in bacteria that produce DNA degrading enzyme. So differentiates DNase positive Staph aureus, Serratia, Moracella cataralis, Micrococcus luteus from DNase negative Staph epidermidis and other coagulase negative Staphylococci and non-pathogenic organism. Let's go to the fourth test for gram-positive cocci, the MSA fermentation. So this uses the agar MSA class, which is both selective and differential. Now the thing about MSA class is that it contains 7.5% sodium chloride, which in most bacteria class, salt is known to inhibit most bacteria. So it inhibits most bacteria but allows salt-tolerant Staphylococci to grow. Meaning class MSA is designed for your Staph species. It also contains mannitol and the pH indicator phenol red. Bacteria that ferments mannitol produce acid, lowering the pH, which changes the medium color from red to yellow. Non-mannitol fermenters grow without a color change. Medium stays red or pink. Here's the procedure class. So inoculate on MSA, incubate, observe for growth and color change. If the medium becomes it grows with a yellow halo, so colonies grow a yellow halo surrounding them, it's positive, meaning the colony or the bacteria is growth tolerant, salt growth tolerant. If it's negative, no fermentation, there would be growth but medium would remain red or pink. If it's no growth, organism is not salt tolerant. So take note of this class. So again, pag positive siya, if it's positive for mannitol fermentation, growth with a yellow halo around colony. So there is salt tolerant siya, and at the same time, it's able to ferment mannitol. But if it's not able to ferment mannitol, no fermentation, growth but medium remains red-pink. No growth meaning not salt tolerant. So ito siya. So this is your salt tolerant that's able to ferment mannitol. Well, itong dito sa part na to, walang colony, walang growth. So meaning unable to, not salt tolerant siya. Usually Staph aureus to. Same with this class, itong mga colonies dito. These are salt tolerant bacteria able to ferment mannitol. Useful for selective isolation of staph Differentiates pathogenic stuff from non-pathogenic. Use again for S. aureus, which can ferment mannitol. So used to differentiate mannitol fermenters, positive with a yellow halo, Staph aureus, from non-fermenters, negative, Staphylococcus epidermidis, Staphylococcus saprophyticus. No growth, most non-staphylococcal bacteria. Let's go to the fifth one, novobiocin susceptibility test. So this one uses the antibiotic novobiocin. It inhibits the enzyme, the bacterial enzyme DNA gyrase. Susceptible organisms would show a distinct zone of inhibition around the novobiocin disc, while resistant will grow up to the edge of the disc. Measuring the diameter of the inhibition zone determines whether the isolate is resistant or susceptible. So here's the procedure class. So you're going to prepare your test organism on a TSA or a MHA. Inoculate the agar surface uniformly. Place a novobiocin antibiotic disc. Incubate the plate at 35 to 37 degrees for 18 to 24 hours. Then after incubation, measure the diameter of the zone. Resistant if the zone inhibition is less than 16 millimeter. Susceptible if the zone of inhibition is greater than 17 millimeters. So for you to better understand, ito siya. So this is an example class of your Muller-Hinton agar plate or your MHA. So you're going to inoculate the organism here. I-spread nyo siya dyan. Then you add the novobiocin antibiotic disc. So ito yung novobiocin nyo. Now, if the bacteria class grows a zone of inhibition, when you say inhibition, it basically prevents the growth of bacteria, meaning wala kayong makikitang colonies. There's no colony present. If the zone of inhibition is less than 16 millimeters, so this is the zone of inhibition. Yan, yung bilog na yan. You measure the diameter. So kung ito yung bilog, ito yung zone of inhibition. If this is the zone of inhibition, you measure the diameter, yung middle part. If the diameter is less than 16 millimeter, you report that as resistant. But if the zone of inhibition class is greater than 16 millimeter, so let's say this is 23 millimeters class, you report that as sensitive, which is usually Staphylococcus aureus, while the resistant is Staphylococcus saprophyticus. So this is a rapid and reliable method for distinguishing between clinically important coagulase negative staph. It is particularly valuable in identifying Staphylococcus saprophyticus, which is a frequent cause of UTI. Ensures correct diagnosis and treatment by preventing misidentification of staphylococcal isolates. Differentiates novobiocin resistant Staphylococcus saprophyticus from novobiocin susceptible Staphylococcus epidermidis and other coagulase negative staphylococci. So do take note of that. Now we have bacitracin susceptibility test. So this is based on the ability of bacitracin, an antibiotic, to inhibit the growth of susceptible bacteria by interfering with cell wall synthesis. So when a bacitracin disc is placed on an inoculated agar surface, susceptible organisms show a clear zone of inhibition. The test is primarily used for the presumptive identification of Streptococcus pyogenes or your group A strep, which is usually bacitracin sensitive. So earlier you had your novobiocin for the staph species class. This time we will use bacitracin. So this is the procedure. So get a pure culture, inoculate on blood agar plate this time. So BAP naman. Place a 0.04 micro bacitracin disc, incubate, examine for the presence. Zone of inhibition present, sensitive to bacitracin. No inhibition zone, resistant. So ito siya. So if you take a look at this class, this is your S. agalactiae, which usually shows a resistant pattern. So ayan siya. Very, very small or no zone of inhibition. But if you take a look at the Streptococcus pyogenes, sensitive siya. has 14 millimeter on a blood agar plate. So provides a simple and rapid method for presumptive identification of S. pyogenes, significant for group A strep class known to cause a lot of diseases such as pharyngitis, cellulitis, scarlet fever, and so on. Helps differentiate between pathogenic strep from other beta hemolytic strep. Differentiates bacitracin sensitive S. pyogenes or your group A strep from other beta hemolytic strep such as the group B strep S. agalactiae. Now let's go to another antibiotic susceptibility. This time si optochin. Now optochin class was previously known also as your Taxopi antibiotic. So the optochin susceptibility test is used to identify Streptococcus pneumoniae. Optochin, as the chemical name ethyl hydroxypyr hydrochloride, interferes with the ATPase and proton pump activity of susceptible bacteria, leading to cell death. Now, when an optochin disc 5 to 6 millimeter containing 5 microgram optochin is placed on a blood agar plate, inoculated with alpha hemolytic strep, S. pneumoniae shows a distinct zone of inhibition around the disc, while other alpha hemolytic strep, such as the viridan group, would show no inhibition. So this is the procedure. So prepare your culture, inoculate on blood agar, then place your 5 microgram optochin disc. Incubate, measure the zone of inhibition. If it's greater than 14 millimeter, using a 6 millimeter disc, sensitive to optochin, meaning it's Streptococcus pneumoniae. But if the zone of inhibition class is 14, less than 14 millimeter, report it as resistant. Most likely, this is a viridans streptococci. So, ito siya. So, on the left side is an example of your viridans strep, Streptococcus mitis, showing a resistant pattern to optochin or your Taxopi. While on the right side is a sensitive result showing a big zone of inhibition susceptible to optochin, meaning this is possibly Streptococcus pneumoniae. So this is a rapid identification for S. pneumoniae, distinguishes S. pneumoniae from other viridans group. Clinically significant because S. pneumoniae is a major cause of pneumonia, meningitis, sinusitis, and otitis media. Differentiates strep pneumoniae from viridans strep, S. mitis, mutans, salivarius, and others. Now let's go to another test, the bile solubility test. So this is based on the ability of bile salts such as your sodium deoxycholate to lyse bacterial cell wall. Streptococcus pneumoniae produces an autolytic enzyme that is activated in the presence of bile salts, leading to cell lysis and clearing of the suspension. So this produces a clear zone class pag positive. Other alpha hemolytic strep, such as your viridans group, do not undergo lysis when exposed to bile salts. The solubility in bile distinguishes S. pneumoniae from other alpha hemolytic streptococci. So this is the procedure. So there are two classes, the tube and the plate. So let's start with the tube method. So label test tube, one for bile and one for saline, which is the control. Add 0.5 ml of 2% sodium deoxycholate solution to the bile tube. Add 0.5 ml of saline to the control tube. Transfer 2 loopfuls of the bacterial suspension into each tube. Add 2 drops of phenol red indicator and adjust pH to 7 if needed. Incubate both tubes at 35 to 37 degrees in a water bath. Observe for clearing every 30 minutes for up to 2 hours. So ito siya, class. So if it's lyse by bile yung solution nyo sa tube, it would become clear. So meaning the bile salts are destroyed or lyse, producing a clear solution. For the plate method, you're going to grow colonies on blood agar. Add 1 to 2 drops of 10% sodium deoxycholate. Then observe for dissolution or flattening of the colonies within 5 minutes. Positive result, clearing of suspension in tube or colony dissolution, S. pneumoniae. No clearing or dissolution, viridans strep. So, ito siya. So, this is the colonies class. If you take a look, they are starting to clear yung paligid niya. So, this is a prime example of a positive result. So, again, this is for S. pneumoniae, differentiating other from other alpha hemolytic. So, bile-soluble Streptococcus pneumoniae, bile-insoluble the Birivan strep, S. mitis, mutans sanguis, and salivarius. Next test is your CAMP test class. So, this is the based on the ability of group B strep, Streptococcus agalactiae, to produce the CAMP factor, which is a diffusible, heat-stable protein. This CAMP factor enhances the lysis of red blood cell by the beta hemolysin produced by Staphylococcus aureus. When group B strep are strict perpendicular to a streak of S aureus on blood agar, the interaction produces an arrowhead-shaped zone of increased hemolysis at the junction, indicating a positive result. So this is the procedure. Inoculate a sheep blood agar plate with a single streak of Staph aureus down the center. Streak the test organism at a right angle to the S aureus streak, leaving about 2 to 3 mm space between the two streaks. So kung ito yung plate nyo, class, so sa center you would streak si Staph aureus. Then dito sa right side, so right at the right angle, mag-i-streak ka ng Streptococcus, but there has to be space about 2 to 3 mm. Then incubate 35 to 37 and examine the hemolysis pattern. An arrowhead-shaped zone of enhanced hemolysis at the intersection of the two organisms is positive, while no enhancement of hemolysis is negative. So ito siya. So here you have, class, your Staph. So ito yung S aureus nyo. Then you streak at a right angle yung suspected strep nyo. So if there's no arrowhead, walang arrowhead dito sa junction, it's negative. But if there's an enhanced hemolysis, katulad nito, producing a so-called a arrowhead hemolysis pattern, it is considered positive. So this is a rapid test for group B strep S. agalactiae, known to cause class neonatal sepsis. So pakitandaan yan, tinatanong yun sa boards. Differentiates group B strep from other beta hemolytic strep which lacks the CAMP factor. Used to differentiate group B from group A, group C, and other beta hemolytic streptococcus. Another test class is the PYR test. So the PYR test is also known as your pyrrolidonyl aralamidase test. Detects the bacterial enzyme L-pyrrolidonyl aralamidase. This enzyme hydrolyzes the substrate L-pyrrolidonyl beta-naphthylamide, releasing beta-naphthylamide. Now, in a color developer, your color developer class is your para-dimethylaminocinnamaldehyde is added. The beta-naphthylamide reacts to produce a bright cherry red color. A positive color changes within a few minutes, indicating the presence of the enzyme. So positive here is a bright cherry red color. So place a PYR disc or strip on a clean surface. Moisten the disc lightly with sterile distilled water. Then using a sterile loop or applicator stick, rub a small portion of the test organism onto the disc. Incubate at room temperature for about 2 minutes. Add a few drops to the color developer, your para-dimethylaminocinnamaldehyde, then observe for the result. Cherry red color within 1 to 2 minutes is positive. No color or only a yellow or orange color is negative. Excuse me. So this is your PYR test. So a cherry red color class after adding your para-dimethylaminocinnamaldehyde would indicate a positive PYR test. So rapid and simple test for specific gram-positive cocci and enterics. Clinically significant because group A strep and enterococci are significant human pathogens. So, ang mga PYR positive nyo class is si Group A, si S. pyogenes, and the Enterococcus, E. fecalis, and E. faecium. PYR negative are Group B, Agalactiae, viridans, and most non-enterococcal Group D, strep. Then we have your bile esculin hydrolysis test. This is based on the ability of certain bacteria to grow in the presence of bile salts and to hydrolyze esculin. So when esculin class is hydrolyzed, it produces the so-called esculitin, which reacts with ferric ions in the medium to form a dark brown to black precipitate. Growth in the presence of bile plus blackening of the medium indicates a positive test. Procedure class. Streak or inoculate a bile esculin agar slant or plate with the test organism. Incubate at 35 to 37 for 24 to 48 hours. Observe the medium. Growth with black or dark brown coloration in the medium. Negative, growth without blackening or no growth at all. So ito siya. So if the medium class, your bile esculin agar still is brown in color, it's negative. But if it darkens class, it means that bile esculin hydrolysis occurred, producing a positive result. So rapid, reliable method for identifying bacteria able to tolerate bile and hydrolyze esculin. Used to identify enterococci and group D streptococci. Helps distinguish these organisms from streptococci that do not hydrolyze esculin in the presence of bile. So bile positive are your Enterococcus, A. fecalis, A. faecium, Streptococcus bovis, the group D. While negative are the viridans, group A, B, and C. Then another test for your gram-positive cocci is the 6.5% sodium chloride growth test. So most somehow similar siya class sa mannitol salt fermentation. So this is based on the ability of your bacteria to tolerate and grow in high salt concentration. So high salt levels inhibit most strep, but enterococci and some salt-tolerant organism can grow in this environment. Growth turbidity in 6.5 sodium chloride broth indicates salt tolerance, while no growth indicates sensitivity to high salt. So you're going to incubate or inoculate your sample or your colony on a 6.5% sodium chloride broth. Incubate at 35 to 37 degrees. Observe the broth for turbidity or sediment. Visible growth in the broth is positive. Broth remains clear with no growth means negative. So ito siya class. If there is the presence of turbidity, it indicates positive. So clear, but these are the positive, meaning salt tolerance siya. So provides a simple and reliable method to separate Enterococcus from non-enterococcal group D. Important class because your Enterococcus are known to cause UTI, bacteremia, endocarditis that are often multi-drug resistant. So differentiates or positive Enterococcus fecalis, Enterococcus faecium. Negative in 6.5% sodium chloride. So that ends the gram-positive cocci test. Okay, let's continue with the gram-positive bacilli class. So there are also tests for your gram-positive bacilli. We're done with the cocci. Now let's go to the bacilli naman class. So the most common test for gram-positive bacilli class is their motility. So this is determined by the bacteria's presence of your flagella. So certain bacilli, Listeria, Bacillus have flagella and are motile, while some, like your Corynebacterium, are non-motile. So motility can be detected by observing bacterial spreading away from the stub, by wet mount under the microscope, or by characteristic patterns such as yung umbrella-shaped motility ng Listeria. So this is the different procedures, or there are two. Let's start with the tube method using a semi-solid agar. So you put or you make a stab in a sim media. So semi-solid motility media by stabbing the center, incubate, and if suspected is Listeria, atrum them. Observe the pattern of growth. If there is diffuse, hazy growth radiating outward from the stub line, forming an umbrella spread-like growth class, this is most likely class a positive result. But if growth confined strictly to the stub line with no spreading, negative. So ito appearance niya. So you would stab the media at the center if it doesn't spread class. So from the center, stab mo siya. If it doesn't spread, stays at the line, negative siya. But if it spreads class, following that so-called umbrella pattern. So ito, katulad ito, this is an umbrella. So most likely, this is your Listeria. And in some cases, overgrowth siya class. There would be an overgrowth wherein the entire medium would become red, signifying growth. Then another one is the hanging drop. So this was supposedly done in the first part of the subject. Prepare a hanging drop or wet mount. Examine under a high power objective. Observe for true directional movement, not Brownian movement. So you would use here class a biconcave slide or a concave slide. So they have a concave well. So you inoculate here the suspension, or you would view here the suspension on a cover glass. Then you would look for directional motility. So used to differentiate a lot of bacteria class, specifically from motile, from non-motile, because a lot of bacteria, such as your Listeria, can cause meningitis and septicemia. So motile organism class, Listeria monocytogenes, producing an umbrella-shaped motility. Bacillus, general motile, diffuse spreading. Clostridium tetani, Clostridium septicum, motile under anaerobic condition. So non-motile are Corynebacterium, Bacillus, Anthracis, and Clostridium perfringens. Take note of that. Now let's go to another test, hemolysis on blood agar. So some gram-positive bacilli class would show the different hemolysis on a blood agar plate. So just a review, pag beta hemolysis, it's complete lysis, alpha hemolysis, partial, gamma, no lysis. So procedure, streak on a blood agar plate, incubate aerobically or anaerobically, examine the plate. If there is clear zone or transparent zone, beta, greenish or brown zone, alpha, no change, gamma hemolysis. So ito siya. So if you streak here like this class and there is clear zone surrounding it, beta. If it's greenish, if there is a greenish clearing at the edge or a greenish zone at the edge of the colony, it's alpha. If there's no change, then it's gamma hemolysis. So it helps in the presumptive identification of gram-positive bacilli. Clinically relevant class in identifying Anthracis or Listeria monocytogenes. So ito yung mga result niya. If it's beta hemolytic class, it's usually Bacillus cereus, Listeria monocytogenes, and Clostridium perfringens. If it's non-hemolytic, gram-positive bacilli, we have your Bacillus anthracis and Corynebacterium diphtheriae. If it's variable or alpha hemolytic, some Corynebacterium species. Occasional Listeria strains. Another test is yung lecitinase test, which uses an egg yolk agar. So this test detects the enzyme lecitinase, also known as your phospholipase C, produced by certain bacteria. Now, lecitinase hydrolyzes the phospholipid lecithin in egg yolk agar into insoluble diglycerides. This results in the formation of an opaque, white to pearly precipitate surrounding the colony. The presence or absence of this opaque zone is used to determine lecitinase activity. So, ito yung procedure niya. So, prepare and dry an egg yolk agar plate. Streak or spot inoculate the test. Incubate the plate at 35 to 37 degrees for 24 to 48 hours. Examine around colonies for opacity. Positive would show an opaque, white, pearly precipitate surrounding the colonies. Negative, no precipitate. Agar remains clear around the colonies. So ito siya. So this is the colony that was strict class. Now if you take a look at the surrounding, there's opacity signifying a positive result. Provides a specific test for toxin-producing gram-positive bacilli used in differentiating pathogenic organisms that produce lecithinase. Clinically important in identifying C. perfringens, which produces lecithinase. Also aids in differentiating Bacillus cereus from Bacillus anthracis. So ito siya, differentiates lecithinase positive perfringens, cereus, and Clostridium bifermentans. Lecithinase negative, Bacillus anthracis, Listeria monocytogenes, and most Corynebacterium species. Then we have your lipase test. So the principle here is lipase detects the enzyme lipase, which hydrolyzes lipids such as triglycerides into two substances, glycerol and free fatty acid. On egg yolk agar, lipase activity appears as iridescent pearly sheen on the surface of the colony. This sheen differentiates lipase producers from non-lipase producing organism. Procedure: inoculate the test organism onto the surface of egg yolk agar. Incubate at 35 to 37 degrees for 24 to 48 hours. Observe the colony. Positive is an iridescent pearly sheen on the colony surface. Negative, no sheen. So itong first picture on the left, this is a negative result. While on the second one, if you take a look at the top class, you would see here a iridescent change in color. So nagiging shiny siya. That indicates positive result. Used to detect lipase producing gram-positive bacilli, differentiate among species of Clostridium and Bacillus. Important virulence factor in some pathogens aiding in tissue destruction. So differentiates lipase positive Clostridium sporogenes, Clostridium botulinum, and some Bacillus species. Negative class are your perfringens, anthracis, and Listeria monocytogenes. Another test is your urea test, which detects the enzyme urease. Now, the urease class is known to hydrolyze urea into ammonia and carbon dioxide. The release of ammonia increases the pH of the medium, causing the pH indicator phenol red to change color. A positive result is seen as a pink, meaning alkaline color, while a negative result remains yellow to orange. So, tandaan class, if it's positive pink color, negative results would remain yellow to orange. Reaction: urea is converted by urease to ammonia and carbon dioxide. So, inoculate the test organism into a Christensen's urea agar slant or into a urea broth. Incubate at 35 to 37 degrees. Broth observe up to 24 hours. Agar observe up to 24 to 48 hours. Examine for color change. Positive is again pink. No color change. Medium remains yellow to orange. So ito siya. If it's yellow or orange class, it's a negative urea test. But if it becomes pink or magenta, positive results. Provides a rapid test for bacteria that produces urease. Helps in the differentiation of gram-positive bacilli and some gram-negative bacteria. Common ang urease-positive bacteria class to cause UTI and contributing to stone formation by alkalinizing urine. So, used to differentiate urease-positive, gram-positive bacilli, Corynebacterium urolithicum, and some Corynebacterium species. Urease negative are your Listeria, Bacillus species, and Clostridium species. Another test is your nitrate reduction test nyo. So, some bacteria would reduce nitrate, NO3, to nitrite, NO2, or further to nitrogen gas or ammonia. The reduction is detected by adding sulfonilic acid and alpha-naphthylamine, which reacts with nitrite to form a red color. If no color develops, zinc powder is added. If it produces a red color after zinc, it is a true negative. Nitrite is not reduced. If there's no color after zinc, nitrite is reduced beyond nitrite. So ulitin ko class the reduction is detected by adding sulfonylic acid and alpha-naphthylamine which reacts with nitrite to form a red color. Now, if there's no color development, let's say nag-negative siya, you add zinc powder. This is done to confirm if it's truly negative. Now, if you add zinc powder and the color still is becomes red after adding it, then it's true negative. But if there's no color after zinc, nitrite reduced beyond nitrite. So take note of that. So inoculate nitrate broth with a test organism, incubate at 35 to 37 degrees, add 5 drops of sulfonylic acid and alpha-naphthylamine. Red color is positive. If no color, add zinc powder. Red color after zinc, negative. No color after zinc, positive for the reduction of nitrate to nitrite. To ammonia, sorry. So ito siya class. So the red color would indicate a positive result after adding the sulfonylic acid and alpha-naphtol. Helps differentiate gram-positive bacilli that may be morphologically similar, important in classifying Bacillus species and some Corynebacterium species. Differentiate Bacillus subtilis. Positive is Bacillus subtilis and some Corynebacterium species. Negative, Bacillus anthracis and Listeria monocytogenes. Gelatin hydrolysis test principle is some bacteria produces gelatinase that hydrolyzes gelatin into amino acid, causing liquefaction. Gelatin hydrolysis is a marker of proteolytic activity. So this is again another test class. It produces a liquefaction of the media. So stab into the nutrient gelatin medium, incubate at 35 to 37 degrees, refrigerate for 30 minutes. Positive, medium becomes liquid. Negative, medium remains solid. So ito siya. So if the medium becomes, so after stabbing, magiging red siya class. If it starts to liquefy, positive siya, meaning na-hydrolyze yung gelatin. But if the media still is solid, negative siya. So detects extracellular protease production used for differentiation of gram-positive bacilli. So differentiates Bacillus subtilis, Clostridium perfringens, which are positive, and negative naman si Anthracis, Listeria monocytogenes, and Corynebacterium species. Then another test is your carbohydrate fermentation test. So bacteria class is known to ferment sugar producing acid, detected by pH indicator and sometimes gas trapped in Durham tubes. So inoculate sugar broth, glucose, sucrose, lactose, mannitol containing phenol red and a Durham tube. Incubate at 35 to 37 degrees for 24 to 48 hours. If the media becomes yellow, it's acid production. If gas bubble in the Durham tube, it indicates gas production. So ito siya. If the media remains red class or pink, negative siya. Not acid producing or not acid fermenting. But if it becomes positive, if it becomes yellow, it signifies that it's positive and it was able to ferment the sugar on the media. Also the presence of bubbles dun sa Durham tube. Ito yung Durham tube nyo yan. It indicates gas production. Used to determine carbohydrate utilization, aids in differentiation of gram-positive bacilli like Corynebacterium, Bacillus, Clostridium, and Listeria. So Listeria is known to ferment glucose maltose. Corynebacterium diphtheriae ferments glucose maltose but not sucrose. Bacillus anthracis, glucose fermenter, sugars with gas. So do remember that. Another specific test class, yung ELIX test. This is designed for Corynebacterium diphtheriae. So it detects the diphtheria toxin, wherein you would use a strip of filter paper impregnated with antitoxin. When toxin-producing C. diphtheriae grows perpendicular to the strip, toxin-antitoxin precipitin line forms. So place the antitoxin-soaked filtered paper on the strip on serum agar. Streak suspected C. diphtheriae isolate at right angle to the strip. Incubate at 35 to 37 for 1 to 2 days. Observe for precipitin line at 45-degree angle from the streak toward the strip. So you add your strip class. Ito yung strip na yan. This has the antitoxin. Then you streak your suspected Corynebacterium. So streak mo siya ng perpendicular. Then after that, you would look for this so-called precipitin lines if positive. So ito siya. Yan yung mga precipitin lines. Now, presence of precipitin lines would indicate a positive result. Distinguishes toxigenic from non-toxigenic C. diphtheriae, essential for confirming pathogenic potential. Positive is seen in toxigenic C. diphtheriae, negative is in non-toxigenic C. diphtheriae or diphtheroids. Another test, Nagler's reaction naman class. This is designed for Clostridium perfringens. So it detects lecitinase alpha toxin activity of C. perfringens on egg yolk agar. So similar siya sa lecitinase test class, produces an opaque precipitate. If antitoxin is applied on one half of the plate, precipitate is inhibited there, confirming specificity. So inoculate on egg yolk agar, streaking across both halves. One side would contain the alpha antitoxin. Incubate anaerobically for 1 to 2 days. Observe. Positive is opaque zone on untreated side, no opacity. Negative, no opacity on either side. So if one side class, so kung ito yung plate nyo, hatiin mo yung sa gitna, separated in the middle, one side you would have the toxin, the antitoxin, while the other half would have no antitoxin. So if there would be opacity on the no antitoxin, positive siya. But if both sides become opaque, negative. No opacity on both sides, negative siya. Then importance, specific diagnostic test for C. perfringens, alpha toxin, confirms pathogenic species in clinical specimen. Differentiates Clostridium perfringens, negative from other Clostridia non-lecitinase producer. Then we have your TSI. So TSI agar contains the following sugar class: the glucose, lactose, sucrose, and other substances such as your ferrous sulfate and phenol red. It detects carbohydrate fermentation, gas reduction, and hydrogen sulfide production. So you're going to stab and streak the slant with the test organism. Incubate at 35 to 37 degrees for 18 to 24 hours. Observe the following. If the slant or the butt becomes yellow, it's acid. If there's gas, cracks or bubbles would be present. Black precipitate would be hydrogen sulfide production. Now, I want you to take note of the following class. In TSI, if it's yellow, it's considered acid. If it's red, it's considered alkaline. Now, they have this so-called shorthand nomenclature. If it's A, if it's yellow or acid, you write it as A. If it's red or alkaline, you write it as K. So let me repeat. If your medium becomes yellow, it means that it's acidic. If it stays red, it's alkaline. Now, meron siyang shorthand. A is for acid, K is for alkaline. Now, this media class is considered a butt and a slant, wherein you would stab and you would streak. meaning you would be showing two results. One result, class, would be the slant, and one result for the butt. Let's say, class, that the slant is red in color, but the butt is yellow. This means, class, that the slant is alkaline, so you write it as K, while the butt is yellow, you write it as A, acidic siya. So K over A. So I want you to think of that because that's important when interpreting the result. So let's go here. In the first tube, class, this is a normal TSI. Kulay red siya. So if you take a look, the slant is red. So that's K over K kasi pareho silang alkaline. You take a look at this second picture, same din. Red slant, red blood, no gas. So K over K. If you take it to the third tube, red slant, yellow butt. So you would write that as K over A. Yellow slant, yellow butt. So this is A over A. And if you take a look, there is cracking of the media, meaning this is positive for gas production. Now here you would have a black precipitate or a blackening of the media signifying hydrogen sulfide production. So when this happens, class, you would always report this as acidic or yellow. You may be wondering, sir, why yellow? That's the standard rule. Whenever there will be blackening of the media, you report that as yellow or A. So you would report it as A over A with H2S production. And there's also cracking, so positive din sa gas. While this one, red alkaline siya, K over A, with hydrogen sulfide and gas production. So that's how you read your TSI. So differentiates gram-positive and gram-negative bacilli with similar morphology. Rarely primarily for gram-positive, but Listeria and some Clostridium show distinct reaction. Differentiates C perfringens, acidic butt, variable slant, gas, no hydrogen sulfide. So may possibility C Clostridium perfringens class would show as A over A or A over K. Alkaline slant naman or butt, no gas. So if it's alkaline, K over K, no gas, no H2S for bacillus. Listeria monocytogenes, A over A, no gas, no hydrogen sulfide production. So growth at different temperature, common to sa Listeria monocytogenes nyo class because they can grow at cold temperatures. Very common sila class in causing diarrhea because it can grow in refrigerator temperature. So inoculate the suspected culture in a broth or agar. Incubate parallel sets at 4 degrees and 35 to 37 degrees. Observe for growth. Growth at both 4 degrees and 37 indicates temperature tolerance. So helps identify Listeria. So growth at 4 degrees and 37, Listeria yung class. No growth at 4 degrees, Corynebacterium and Bacillus species. And the last test for gram-positive bacilli class is yung reverse camp test. So if you still remember yung camp test nyo for the gram-positive cocci, meron naman this time yung reverse. So the reverse camp test detects the production of lecitinase by Clostridium perfringens. Unlike the standard camp test, which enhances hemolysis, the reverse camp test shows inhibition of hemolysis. So when C perfringens is strict perpendicular to a beta hemolysin, a group B strep S agalactiae, the alpha toxin of C perfringens neutralizes the beta hemolysin. This results in characteristic arrowhead-shaped zone of hemolysis inhibition at the junction of two organism. So this time class, gagamit ka ng group B strep CS agalactiae. So prepare a blood agar plate, streak a straight line of strep agalactiae down the center, then streak perpendicularly your C perfringens. Incubate anaerobically and examine the plate for the pattern of hemolysis. So it provides a simple, inexpensive, and rapid confirmatory test for Clostridium perfringens, useful in routine diagnostic bacteriology, often used in conjunction with other confirmatory tests. Differentiate positive reverse camp test Clostridium perfringens, negative other Clostridium species, and unrelated anaerobes. So this is the reference use. Thank you.